Assalamualaikum and good morning everyone. My name is Norshazwina Binti Ahmad Shahir. Matrix number 73668 from subgroup 3. So I will begin this presentation with an overview of this poster. So basically this poster is about how biopesticides can improve biopesticides improve the value of home compost. So biopesticide that we'll be using is the Bacillus thuringiensis. It's a common microbial biopesticide and a gram-positive bacteria that produce a crystal protein that is harmful to numerous insect pests when being eaten. So next, the objective of this research is about to determine the viability of producing Bacillus thuringiensis enriched quality home compost from bio-waste using an inoculum, and the second objective is about to present a strategy to produce enriched home compost with biopesticides properties through a simple and low-cost process. Next, we will proceed with how this research will be conducted.
So hello everyone, my name is Haziq bin Hamidun with the matrix number of 73621. So I will be presenting on the methodology part. So the first step is the preparation of the bacteria inoculum. So the bacteria pearl will undergo incubation, grown in the nutrient broth and centrifuge. And next, the biomass will be incorporated with the fruit and vegetable leftovers to be used for the home composer. So for the home composer, they actually make two identical home composing bin, which will label HC1. They have the inoculated bacteria, and HC2 without the inoculated bacteria. Next, they proceed to the analytical method, in which scanning electron microscopy were used to identify bacteria toxin morphology, biological stability, and dynamic cumulative respiration index for 4 days. Next, they move on to the toxicity test, and this was carried out by CREF, which is the Universitat Autonoma de Barcelona. They observed the growth of the Phosphomia candida using the image processing software. So next, they proceed towards the microbial analysis and also molecular identification. So the genomic DNA was extracted, and the region of interest was amplified to be identified of the isolates to be compared using the data from gene bank. And lastly, they do the DNA extraction and sequencing. The samples for both composts were processed to achieve DNA extraction using PowerSol TM DNA isolation kit, and also the BLAST tool were used to check whether the sequence DNA are available in the gene bank. So next, we will proceed to the result and discussion.
So hello everyone, my name is Irfan Charles, matrix number 73581. So I will be presenting regarding our result and discussion. After 148 days, there were no significant changes between the two composts in terms of organic matter and nutritional content. Both composts evaluated were declared mature and safe for use in soil. The germination index, GI, was also determined to evaluate the compost maturity. All samples showed GI over 100%, indicating that they are all mature with no phytotoxic substances, demonstrating that adding Bacillus thuringiensis to the home composting had no effect on the final compost quality. The relative abundance of both of the composts were also quite similar, indicating that Bacillus thuringiensis had no effect on the global structure of the dominant microbial community, though it did structure the species of the prokaryotic community within each phylum. So further research work on the Bacillus thuringiensis growth may be needed to confirm the self-sufficiency of the process. However, present studies suggest that there will be successful. So that is all for the result and discussion.
So as conclusion, there is an effective strategy that being applied to produce the Bacillus thuringiensis enriched compost from home compost by using a solid Bacillus thuringiensis inoculum. And in contrast, there is also a home composting bin without the inoculation. And as conclusion, there is no difference between these two composts. And although Bacillus thuringiensis inoculation did not show major changes in microbial diversity or global composition of the microbiological community, it can alter the quantitative structure of each phylum, causing a redistribution of relative abundance of species. So that's all from us. Thank you for watching.
Thank you.